Welcome to our OpenSim webinar. My name is Jennifer Hicks, and I'm the OpenSim R&D Manager and Associate Director of the NCSRR, and I will be serving as the moderator for the webinar today. I'm pleased to welcome today's presenters from the University of Delaware, including Jill Higginson, Brian Knarr, and John Ramsay. OpenSim is a freely available software application for visualizing musculoskeletal structures and simulating movements of humans and animals. The application includes tools for general-purpose inverse dynamics, optimization to estimate muscle and joint forces, methods to create simulations for motion capture, and tools to analyze and visualize the results of simulations. The first goal of our webinar series is to showcase cutting-edge research that is being performed with OpenSim. OpenSim is also a growing and geographically diverse community of users, and thus the second goal of our webinar series is to provide an easy platform for the OpenSim community to communicate and collaborate. Now, before we get started, I have a few reminders about the webinar format. Questions will be addressed at the end of the presentation using the Q&A panel. And if you need additional technical help, you can consult the guide on our website. And now I'd like to introduce our speakers. Dr. Higginson is an Associate Professor in the Department of Mechanical Engineering and Associate Director of the Biomedical Engineering Program at the University of Delaware. Her doctoral degree is from Stanford University in Mechanical Engineering. Ongoing projects supported by the NIH target abnormal muscle control strategies in stroke and osteoarthritis. Brian is a postdoctoral researcher with the Delaware Rehabilitation Institute at the University of Delaware. His doctoral degree is in biomechanics and movement science from the University of Delaware, and he has a master's degree in mechanical engineering. His research interests include experimental and simulation methods to advance gait rehabilitation in neurological populations. John Ramsay is a PhD student in the Biomechanics and Movement Science program at the University of Delaware. He has a bachelor's degree in biomedical engineering and a master's degree in mechanical engineering. His research involves identifying morphological changes and compensatory strategies in post-stroke individuals, and using musculoskeletal simulations to aid in post-stroke rehabilitation. In their talk today, they'll explore when and why you need subject-specific muscle parameters in individuals who have suffered from a stroke. Take it away, Jill, Brian, and John. Thank you for the opportunity to present our work. Here in the Neuromuscular Biomechanics Lab at the University of Delaware, we're interested in muscle coordination of normal and pathological movement. As engineers, we work closely with physical therapy to understand movement abnormalities, identify underlying deficits, and apply novel therapeutic regimens in individuals with knee osteoarthritis and post-stroke hemoptysis. Typically, we study gait using standard motion capture techniques on an instrumented treadmill. This data is used to create musculoskeletal models and simulations using OpenSim. By using coupled experimental and computational approaches, we hope to identify root causes or underlying mechanisms responsible for movement abnormalities and understand and eventually predict results of therapeutic interventions. And because musculoskeletal models generate vast amounts of data, we need to consider what parameters are most relevant for the question and population studied. In some cases, the use of general parameters may be sufficient. However, as we apply modeling techniques to pathological conditions, we are often dealing with unique subject characteristics. And with these populations, it may be useful to create models that specifically represent a group of individuals or a single individual. As part of an NIH-funded study, we have been collecting an array of experimental data related to muscle strength on individuals who have experienced a stroke and are using this to build subject-specific models and simulations. Ultimately, we hope that this information will be used to guide individualized rehabilitation protocols. Our goals today are to first describe our experimental approach for strength assessment using indicators of muscle atrophy and activation. And second, to incorporate these metrics into a subject-specific simulation to identify differences in muscle coordination patterns. To address this first objective, John Ramsay will motivate and discuss our approach to study atrophy. Thank you, Jill. Here in the United States, stroke is a leading cause of long-term adult disability. A common impairment for chronic stroke survivors is muscle weakness, which is evident by a decrease in force during a paretic limb maximal volitional contraction. Additionally, muscle weakness has been shown to be a limiting factor of gait performance. Therefore, it is a goal of our lab to not only measure post-stroke muscle weakness, but to also identify and understand the role of each contributing factor to post-stroke muscle function. Post-stroke muscle weakness has been attributed to three major changes in the neuromuscular system: muscle activation impairment, muscle atrophy, and overall muscle composition.
Post-stroke activation impairment may arise from a decrease in firing rate for each motor unit, a decrease in the number of functioning motor units, and or the ability to activate individual motor units. Muscle atrophy can be observed through changes in muscle mass, cross-sectional area, or muscle volume. Finally, muscle composition changes may be evident in muscle stiffness or spasticity, as well as a shift in muscle fiber types. The relationship between these factors following a stroke and an individual's ability to compensate for these changes is unclear. The objective of this next study was to determine post-stroke muscle volume changes using magnetic resonance imaging. To measure muscle volume changes, 11 post-stroke adults were included in this study. Axial T1 weighted magnetic resonance images were taken in a 1.5 Tesla scanner. 5 overlapping scans span the entire lower leg region from the ankle to the iliac crest. These overlapping scans were needed to minimize any artifact that may occur during a long spatial scan. This scanning protocol was advantageous for us because it allowed us to scan the entire lower limb from the waist down in just 30 minutes, as well as visualize each individual muscle in vivo. For all subjects, muscles were digitally reconstructed using the iMod software package. Each individual muscle boundary for both paretic and non-paretic sides was identified and manually traced over the entire length of the muscle belly. In the figure below, you can see 3 plantar flexor muscles outlined on both the paretic and non-paretic sides. When measuring muscle volume, one method would be to generate a surface mesh of the muscle faces, seen here, and then calculate the volume from the surface area. However, since we are interested in the active force generating potential of a muscle, we prefer to remove any non-contractile tissue from the muscle volume calculations. To do this, a pixel threshold representing fatty infiltrate was determined for each individual by visually inspecting each scan. In the figure to the right, the same images are displayed on top of each other. However, in the bottom figure, you can see the non-contractile tissue or intramuscular fat identified. Cross-sectional areas were adjusted for non-contractile tissue by subtracting out the fatty pixels, and then volume is calculated by summing the adjusted areas over the length of the muscle and then multiplying by the slice thickness. Muscle atrophy was quantified by comparing the phoretic and non-phoretic fat-adjusted muscle volumes using a percent difference. Overall, most muscles were significantly lower in volume on the phoretic side. Averaging across all muscles, the phoretic muscles were 23% smaller than the non-phoretic muscles. In the shank region, the phoretic muscles were 20% smaller, and in the thigh region, the phoretic muscles were 24% smaller. To the right, you can see muscle volumes for the dorsiflexor, plantar flexor, knee flexor, and knee extensor muscle groups. In yellow are the phoretic muscle volumes, and in blue are the non-phoretic muscle volumes. When we focused on the plantar flexor muscles, we noticed that there is a large difference in muscle atrophy when comparing between individual muscles. The soleus atrophies approximately 12% on the phoretic side, whereas the two heads of the gastrocnemius atrophy twice or even 3 times as much. This suggests that the gastrocnemius atrophies preferentially over the soleus. The soleus, which consists primarily of type 1 muscle fiber, has a lower activation threshold and fatigues less easily than the gastrocnemius. The gastrocnemius may atrophy more because it has a higher percentage of type 2 muscle fibers. This preferential gastrocnemius atrophy may be a key limitation in swing initiation during post-stroke gait, and also suggests that there is a need for muscle models that describe different fiber types to be used in populations where fiber type could be affected. Another interesting finding was found when looking particularly at the tibialis anterior. For this muscle, there was only a 4% decrease in phoretic muscle volume, which suggests that the tibialis anterior atrophies the same amount on each side. In post-stroke gait, dorsiflexor muscle weakness commonly manifests itself as foot drop. This finding suggests that dorsiflexor muscle weakness may be attributed to neurological or muscle composition changes rather than structural changes. So to further our discussion on post-stroke muscle activation deficits, I will now turn the microphone over to Brian.
Thanks, John. So for the first portion of this webinar, John discussed how atrophy can be affected in post-stroke individuals. And for my first portion, I'll be discussing how the work we've done here at UD in quantifying a second source of muscle weakness seen post-stroke, namely activation impairment, and how we quantify this using the burst superimposition technique. Using the burst superimposition test, we are able to obtain an estimate of several values to describe the force-producing capability of a muscle or muscle group. First, we can measure the maximum volitional force that a subject can produce, or MVC. Additionally, we can estimate the maximum force generating ability, seen here as MFGA, and the central activation ratio, or CAR, of a muscle or muscle group. We define MFGA as the maximum force produced by a muscle or muscle group with full activation, and we define CAR as the level of volitional activation a subject can reach in a muscle or muscle group, ranging from 0 or no activation to 1 or full activation. To perform the burst superimposition technique, we use a dynamometer such as a King-Tom or Biodex to measure joint torque. During testing, we deliver electrical stimulation to a muscle group consisting of a 600 microsecond pulse duration, 100 millisecond train duration, 135 volt train of stimulation, delivered at 100 Hertz. During the testing, we stabilize the subject's shoulders and torso to minimize body movement that can augment the force delivered to the force transducer. Subjects are encouraged to produce their maximal effort during the test through verbal motivation. For those of you who are unfamiliar with the burst superimposition test, the graph on the right represents a typical force trace produced by an individual post-stroke during the test. To begin the test, we deliver an initial pulse of stimulation. Following this initial pulse, the subject has 5 seconds to produce their maximum volitional force, labeled as F-ball. At the end of the 5 seconds, the subject receives an 11 pulse burst of stimulation on top of their maximum volitional contraction. The additional force produced by the stimulation can be seen on the graph as F-stim. From this data, we are able to calculate both the MFGA of the muscle group as well as the individual's maximum volitional activation, or CAR, of the muscle. MFGA is calculated simply as the sum of the volitional force produced by the subject and the additional force produced by the stimulation. The central activation ratio, or maximum volitional activation, is then calculated as the ratio of the volitional force produced by the subject and the MFGA of the muscle or muscle group. While the burst superimposition technique performs very well at high levels of volitional activation, error in the test increases as volitional activation decreases. In particular, previous work in our lab has shown that the burst superimposition test tends to underestimate the MFGA of muscle as volitional effort decreases. This is due to the fact that the burst of stimulation delivered to the muscle during the test is not strong enough to fully activate a partially activated muscle. The simple solution to this problem would be to simply increase the level of stimulation delivered. However, a longer, more intense stimulation is generally not tolerable. As an alternative, we have developed an adjustment equation for the burst superimposition test to increase the accuracy of this test in the plantar flexor muscle group with low levels of volitional effort. We collected data on 13 young, healthy subjects to develop the equation, with each subject targeting 25, 50, and 75% volitional effort. Additionally, we had a subset of these subjects perform the test at very low levels of 5 to 10% volitional activation. Error was calculated relative to a maximum effort trial in which the volitional effort during the test was greater than 95%. From the initial results on the left, error reached upwards of 50 to 60% underestimation of the measured MFGA at low levels of volitional effort. Using this data, we developed an adjustment equation to minimize the least squares error in the initial 13 subjects, and then validated this equation by collecting data for an additional 7 subjects, which we've seen in red. On the right graph, you can see the results of this adjustment equation compared to the original results on the left. Using this adjustment allowed us to obtain a prediction of MFGA that was insensitive to the level of volitional activation during the burst superimposition test. This is especially important in populations that may have impaired muscle activation, such as in individuals post-stroke. Now that we had a reliable method for assessing the maximum force-generating ability of muscle, we wanted to compare the results of the burst superimposition testing to the muscle volumes obtained through MRI. In particular, we wanted to assess the ability of MRI to estimate MFGA in a post-stroke population. To accomplish this, we used our burst superimposition test to obtain the relative MFGA between the paretic and non-paretic muscles of an individual post-stroke. As John discussed earlier, the maximum force-generating ability of a muscle can be influenced by both atrophy and muscle composition. However, MRI generally describes only the size of a muscle, and therefore the relative atrophy of a muscle between paretic and non-paretic size. Given this, we hypothesized that while MRI will show volume deficits in the plantar flexor muscles of the paretic limb, it will not provide an accurate estimation of the relative MFGA between limbs compared to a more direct measurement such as the burst superimposition technique. To perform this study, we collected data on the plantar flexor muscle group of 17 individuals post-stroke. We collected muscle activation and MFGA data using the burst superimposition test I just described, and we collected data on muscle volume atrophy using the MRI method that John described earlier in the talk. From the burst superimposition data, we were able to compare the paretic side and non-paretic side muscle performance. In this figure, you can see the maximum volitional force, maximum force-generating ability, and central activation ratio of the paretic limb in gray and the non-paretic limb in white. Overall, we saw that the non-paretic plantar flexors had greater volitional force, and maximum activation during the test when compared to the paretic limb. If we then take the ratio of the paretic and non-paretic values in this graph, and also consider the ratio of the paretic to non-paretic muscle volume that we obtained through MRI, we get the following figure. From this figure, we can see that the volitional force produced on the paretic limb is only 40% of the non-paretic limb. This means that these individuals can generate less than half the force on the paretic side compared to their non-paretic limb. If we look at the MFGA of the two sides, we see that if the subject was able to fully activate their muscle, the paretic side is only capable of producing approximately 65% of the force that the non-paretic muscle could produce. In other words, the paretic side is approximately 35% weaker than the non-paretic muscle. When we compare this value to the relative atrophy between paretic and non-paretic limbs, the MRI data suggests that the paretic muscle is about 80% the volume of the non-paretic muscle. This means that atrophy can explain approximately 20% of the total 35% paretic weakness that was measured, leaving 15% of the relative paretic side weakness unexplained by atrophy. This suggests that MRI volume data does not suffice for determining overall muscle weakness post-growth, and that additional changes in muscle composition are likely occurring which contribute to weakness. So from the results of these studies, we've determined that the burst superimposition test is able to obtain an estimate of both the level of activation impairment in a muscle group, as well as the overall weakness in a muscle group in the form of maximum force generating ability. In general, this method appears to be a simple, low-cost, and effective way to obtain subject-specific data on muscle performance. So now that we have a method for obtaining subject-specific data on muscle performance, the next step is to determine whether including this data into musculoskeletal models will have a meaningful impact on simulation results. For this, we wanted to build and compare the results of simulations of individuals post-growth, both with and without subject-specific muscle information. For this simulation study, we collected data on 4 subjects post-growth, ranging from ages 54 to 74. All subjects were between 16 and 14 months post-growth and had a wide range of walking function, with self-selected walking speeds ranging from 0.18 meters per second to 1.04 meters per second. Subjects' lower extremity Pugel-Meyer score, which is a clinical assessment of motor performance, ranged from 15 to 27 out of a possible 34. First, we collected motion capture data on our 4 subjects. We captured the kinematics and kinetics of these individuals while walking at a self-selected speed using an instrumented propelled treadmill and 8-camera motion capture system. We collected the subject-specific force and activation data for these 4 subjects on 3 muscle groups using the burst superimposition technique. The plantar flexor muscle group was collected with the subject supining and their ankle at 0 degrees of plantar flexion. The dorsiflexors were collected with the subject supine and their ankle at 15 degrees of plantar flexion. And the quadriceps were collected with the subject seated upright with their hip at 90 degrees of flexion and their knee at 60 degrees of flexion. Additionally, we applied the adjustment equation for the plantar flexors as well as a previously developed adjustment equation for the quadriceps. Next, we needed to convert the results of the burst superimposition testing into parameters that we could use in the model. For the maximum volitional activation, we can simply set the maximum activation of the muscles in the model equal to the maximum activation, or CAR, that we measured from the burst superimposition test. This value was prescribed in the control constraints file, or alternatively could be prescribed through the GUI as a value for the max node variable, as you can see in the example on the bottom of the slide. Next, we needed to convert the results of the burst superimposition testing for MFGA. To convert the measured MFGA for the burst superimposition test to model parameters, we need to make a few assumptions to relate the model to our actual subject. First, we started with the condition that the maximum torque generated by the muscle group in the model would be equal to the maximum torque that was measured by that muscle group during the burst testing. Second, we assumed that the muscle moment arms are the same in the model and in the subject. And third, that the ratio of individual muscle forces that contribute to the total torque is the same in the model and the subject. Using these 3 assumptions, we were able to calculate a scaling factor for the maximum isometric force of muscles in the model to represent the measured values from the burst testing. The value for maximum isometric force of a muscle is prescribed in the model file as maximum isometric force variable, as you can see in the example on the bottom of the slide. To generate our models, we used the OpenSim software package to create 3D dynamic simulations using our experimental data. Our model included 23 degrees of freedom and 54 muscle actuators. On the bottom of this slide, you can see a flowchart of the steps we followed through OpenSim to obtain our results. First, we converted our data from motion capture collection into an OpenSim compatible format. This required the generation of 3 files per subject. 2 files containing marker trajectories for the captured motion captured by our motion capture system, one for a static trial and one for a walking trial, and a file containing the ground reaction forces and moments and center of pressures during the walking trial obtained from the force plate data. Next, we performed 3 steps in OpenSim to prepare our model before predicting muscle activation. First, we used the model scaling tool to alter the model's size and mass to more closely match our subject. This is done by using marker locations from the static motion capture trial to scale individual segments of the model. Next, we performed inverse kinematics to calculate a set of motion coordinates for the model, which best matches the experimental markers for each time step. This best match is determined by minimizing the sum of weighted squares errors between the model and experimental data. Third, we ran residual reduction analysis to minimize the effects of modeling and marker data processing errors. More specifically, RRA alters the torso mass center of a subject-specific model and permits the kinematics of the model from inverse kinematics to vary in order to be more dynamically consistent with the ground reaction force data. After residual reduction, we generated 2 versions of the model per subject. One model uses the generic muscle parameters that are defined and come default with the model. And a second model uses the new subject-specific muscle parameters for maximum activation and maximum isometric force that were determined using the burst superimposition data. Finally, we ran computed muscle control to compute the set of muscle excitations that would drive the model to reproduce our experimentally measured motion. From the results of CMC, we calculated the average force and activation of phoretic side muscles in the model over 2 time periods, the full gait cycle and the double support phase of gait when the phoretic limb is the trailing limb. Because we have a limited sample size in this study, we assigned thresholds to determine whether a meaningful change in muscle force or activation occurred with the inclusion of subject-specific muscle parameters. These thresholds are a change of greater than 5% in average activation or a change of greater than 50 newtons of average force over the gait phase of interest. Additionally, we require that this change occur in at least 3 out of the 4 subjects to qualify as a meaningful change across the subjects. To further assess the results of our subject-specific simulations, we compared our results to EMG collected during the motion capture collection. This comparison would allow us to determine whether the changes in muscle coordination as a result of subject-specific parameters are more representative of the subject's actual coordination pattern. We collected EMG for 9 muscles on the phoretic limb, seen on the table on the right, and compared the onset-offset timing of the EMG signals and model predicted activation. Due to the variable nature of EMG in the post-stroke population, we determined the onset and offset of the muscle activations manually. We then compared the EMG and model predicted signals, by calculating the percent agreement over the entire gait cycle. As an example of how we calculated percent agreement between EMG and model activation, the figure on the bottom left shows the on-off state of EMG and simulation predicted activation per muscle over the gait cycle. The EMG signal in blue is on from 0 to 25% of the gait cycle, while the model activation is on from 0 to 50%. The 2 signals are in agreement from 0 to 25% while they are both on, and from 50 to 100% while they are both off. Conversely, the signals are not in agreement between 25 and 50% of the gait cycle. This example would result in a 75% agreement between the 2 signals. In this table, you can see the values of the scaling factors for maximum isometric force and maximum activation used in the subject-specific models for the phoretic limb. Overall, we saw a greater maximum force in the quadriceps, but a much lower maximum force in the plantar flexors, with maximum isometric force ranging from only 15 to 33% of the generic model force. For the dorsiflexors, only one subject was able to volitionally activate their muscles during burst testing, and had a maximum isometric force of 5% of the generic model. All muscle groups exhibited activation impairment during the burst superimposition testing, with the quadriceps and plantar flexors ranging from 16 to 61% activation. For the 3 subjects who could not volitionally activate their dorsiflexors during testing, the maximum activation was set to 2%, the minimum activation for muscles in OpenSim. The figure on the right shows the changes in average muscle force that were observed when including subject-specific muscle parameters. Overall, we saw that 10 muscles showed a meaningful change in force over both the full Gait cycle and the double support phase of Gait, with increased force from the hip flexors and knee flexors, and decreased force from the plantar flexors and dorsiflexors. Overall, we saw a similar pattern in muscle activation and found that a total of 9 muscles showed a meaningful change in both activation and force with the inclusion of subject-specific parameters from the burst testing, including increases again in the hip flexors and knee flexors, and knee extensors, and decreases in the ankle and plantar flexors and dorsiflexors. In general, the increases seen in knee and hip flexor activation are consistent with previous simulation and clinical studies, mainly with hip and knee flexor muscles compensating for plantar flexor weakness. When comparing the EMG and model results before and after including the subject-specific muscle parameters, we found several notable changes. Specifically, including subject-specific parameters resulted in improved agreement with EMG for the plantar flexors and knee flexors. This improvement seen in the knee flexor is notable as no subject-specific information was included for this muscle group, yet improvements were still seen in knee flexor timing. We did see poor dorsiflexor agreement for the subject-specific model. However, this is likely because 3 of the 4 subjects had no volitional activation for the dorsiflexors during our burst testing, so our subject-specific models had no dorsiflexor activation. However, EMG was still measurable during Gait for these subjects. From this study, there were several interesting limitations and findings. First, our subject-specific data from burst testing was limited in scope to only the MFTA and maximum activation of the muscle. If we recall back to our 3 main contributors to weakness, it is likely that additional muscle parameters such as optimal fiber length, tendon slack length, or passive properties of the muscle tendon unit could have an effect on the model results. An interesting finding which highlights the passive properties of the muscle tendon unit was seen at the ankle. In our subjects, there was extreme weakness in the plantar flexors and dorsiflexors, and during the paretic push-off phase of Gait, the model was actually unable to generate the joint torque required using muscle forces alone. As a result of this, model reserve actuators were needed to generate the rest of the joint torque required. And for some simulations, this reserve actuator torque peaked at a value of greater than 20 Newton meters. This is interesting, particularly because we also observed large amounts of baseline or resting plantar flexor torque when these subjects were undergoing our burst superimposition testing. This suggests that these subjects are generating a much larger amount of passive force, which would likely play a significant role during walking, and this may have been captured by our model reserve actuators. Finally, our testing involved only isometric muscle performance, and it's possible that our performance may differ based on task. The inability of our subjects to volitionally activate their dorsiflexor muscles during our supine isometric testing is one example of this, as we did measure muscle activity during walking through EMG, and it's possible that the dorsiflexors are more easily activated during a dynamic task. So to sum up our talk today, this is the first study to create musculoskeletal simulations of individuals post-stroke with subject-specific muscle force and activation data. While MRI showed volume deficits in the plantar flexor muscles of the paretic limb, we found that it did not provide a complete description of the overall weakness experienced in individuals post-stroke. Alternatively, we found that the burst superimposition technique was a relatively easy and cost-effective method for estimating both the overall weakness and the maximum volitional activation of a muscle group. Importantly, this study suggests that subject-specific isometric force and activation data may affect the accuracy of model predictions and should be used when building musculoskeletal models of individuals post-stroke. However, there remains many more properties of muscle that could further influence model results, and these properties warrant further study. Finally, I'd like to acknowledge our funding sources and collaborators for these studies, and we'd be happy to take any questions at this time. Thank you.
So Jill, Brian, John, that was a really interesting talk, a really interesting set of studies. We'll go ahead and get started with the Q&A session now. All of the questions will be text-based. So to ask a question, find the Q&A panel in your WebEx controls and type in your question and make sure you select to ask all panelists. So while people start entering their questions, I'll go ahead and ask the question myself. So you saw for the dorsiflexors that there was, during the MVC tests, there was very little to no volitional control, but they were active during Gait. Do you guys have any ideas for incorporating the EMG during Gait with what you measure from those MVC tests to kind of get a better picture of the capabilities of a particular subject?
Yeah, I think that's a great thing that we need to figure out. I think one of the big problems when working with this population is their ability to activate muscle can be highly variable depending on how they're moving. I think the fact that we see EMG during Gait or a dynamic task suggests that maybe if we're doing testing that we could use a moving task rather than an isometric task, right? So if we could do what's the word? Isokinetic task rather than an isometric task, then maybe we could simulate the motion during Gait and get a better way of testing within the dynamometer. The big problem, of course, if we just use EMG during Gait to help drive our model is we don't have any idea of how much strength that muscle really has overall. So it's hard to parameterize that correctly.
Right. I guess you'd have to use the Gait. You could use Gait or EMG during dynamic tasks to supplement the burst superposition technique that you're using.
Right. But if we don't, the key point is if we don't have an idea of the overall strength of the muscle, then a normalized value such as EMG, it's hard to actually know how much force that relates to.
Right. Okay, so the next question is from Ching Sai. He says, thank you for the talk. What factors may have contributed to the difference in force deficit estimated from the MFGA and MRI measured volume?
So I think, if I understand that correctly, are we talking about that change in 15% between volume and MFGA?
Yeah, I think so.
So, I think we highlighted this a little bit, but the change in muscle composition. So are there changes in the, Brian mentioned the passive components of the muscle. Also, do the fiber lengths change? Are the pronation angles of the muscles, are they changing? Do the types of the muscle fibers themselves, do they convert from type 1 to type 2? Or, I think all of these are, areas for further study and trying to understand where that extra or that lack of 15% is coming from.
Mm-hmm. Okay, thanks. The next question is from Megan Vitt. Can you explain how you normalize the EMG on the non-paretic and paretic sides? Additionally, did they have the same resting EMG on both sides?
So to answer the second part first, I'm not sure if they had the same value of resting EMG, but that resting EMG was taken out of the baseline. So to normalize, we simply used within trial normalization. So the maximum that they experienced within a trial was set to 1, and that baseline that we take out is 0. So a pretty standard way of normalizing.
Okay. And then did they have the same resting EMG on both sides?
I can't recall whether they were the same number, but we did take out the baseline for the trial EMG that we used to normalize.
Okay. Now a question from Bart Bolsterly. He was wondering, he thinks you said that you use subject-specific moment arms, and if so, how did you determine those?
So we did not use subject-specific moment arms. That's one of the key limitations, and another thing that could affect how well our values go into our model. So one of the key limitations and assumptions we made in order to get from our burst testing results to that value of maximum isometric force was that the model moment arms, after that model is scaled to subject-specific size, is an accurate representation of what our subject's moment arms actually are. So that's obviously a key limitation to what we've done. And if we have quick and easy ways to get those moment arms, that would be fantastic. But for the purposes of this study, we did not do that.
Okay. But an interesting area to explore in the future. So now a question from Jeffrey Hansfield. He says, determination of non-contractile tissue in MR images will be sensitive to your threshold value for pixel intensity. What criteria did you use for selecting this threshold?
So yes, that is definitely a limitation in the MR methods that we used. So the actual process of doing that was through visual inspection. So each limb was scanned at the same time, and we looked at essentially each scan, and as we subtracted, they look like, the little white pixels, until we got a value, or until we could visually see that the white was being removed and the gray muscle tissue was being left there. And it was, and this was done across all scans. So if the value was, say 400, it was 400 for that individual person. It was 400 for the whole scan movie.
Okay. Another MRI type question from Katie Reed. She says, great talk. How much of the volume differences that you observe between paretic and non-paretic individuals is due to fatty infiltration, and how much is due to volume differences?
Okay, so first of all, just to clarify, the volumes that were reported in this talk were already were presented that had already had the fat subtracted out of it. So I think her question is then, how much fat did we see? And this varied for each muscle. And offhand, I don't have the exact numbers, but I believe it's somewhere between, say, 20 and 40%. You can look up the actual results though in the paper that were in one of the slides.
Okay. Now a question from Kataro Sasaki. What was the deviation or difference in max isometric force between your estimation and the method where you multiply muscle-specific stress and CCSA?
That's a great question. So we didn't actually do that other PSA method, because when we looked at our comparison, we took the ratio of the paretic and non-paretic limbs. And as part of doing that, some of the specific properties that would go into getting physiological cross-sectional area from just a standard cross-sectional area, such as pronation angle, by dividing the paretic side and the non-paretic side, we make the assumption that that say the pronation angle is equal on both sides, and then therefore cancels out. So that's obviously a key assumption that we made in our ratios. But the reason we did that is because we didn't have a good estimate of what the pronation angle really is. And as we identify through our limitations in both our modeling and most of our work, is that if we can get more information on pronation angle for the paretic side and the non-paretic side, which may very well be something that changes significantly due to a stroke, that would help us in predicting our values much better. But without that information from our standard MRI procedure that we use, we were unable to actually make that direct comparison. So we used the ratios instead.
Okay, thanks. I have a question now. I'm wondering, for the individuals with stroke and their paretic limbs, how much similarity was there within that group? So, for example, if you didn't collect the burst superimposition data and the MRI images, could you have a stroke model that you could use in another lab where you have individuals with a similar pathology?
Mm-hmm. That's a very good question. So we do see a rather wide range of function, and I think I tried to highlight that slightly with the 4 subjects that we selected. Their walking speeds were, I think, around 0.3 up to 1 meter per second. And when we looked at the scaling factors that we calculated, they range from 16% up to 61%. So while there's a wide range, in general they are impaired. If you're looking for a very subject-specific model, I think it's necessary to have that subject come in and actually parameterize what their weakness is on an individual basis. But it seems like for stroke overall, there tend to be underlying impairments that are common. So the plantar flexors and the dorsiflexors tend to be rather impaired, a bit less so on the quads and the hamstrings. But I would say if we could take, like you probably suggest, an average of several subjects or 10, 20 subjects, you could probably get something that would work for a general case. But I would caution using that on a specific subject for an intervention, because it's very possible that in some way that person will differ. So we could get good global ideas of how things may change using a more generic simulation, or, as you suggest, using stroke-specific generic properties. But I would caution using that to prescribe any sort of intervention or rehabilitation, because they're probably all different, at least through our experience here.
Mm-hmm. Thanks. That good answer. We have time for a few more questions, so everyone get your questions in. We have another question from Bart Bolsterly, and he says, in a similar study on PCSA scaling of the shoulder model, he found that the total muscle volume differed substantially between subjects, but that the distribution of this volume over muscles was very consistent. Did you find something similar for the leg?
Distribution. So if we look at each individual muscle group, there was not a similar trend. Some groups were affected more than the others. And across the board, looking at the individual muscles, it was all over the board. I think that's.
What about in the non-paretic limb, or did you have normals as well?
We did not have we did not have healthy. We just looked at the stroke subjects.
Right. Was the distribution different in non-paretic limb? Did you look at that?
No, we didn't look at any of that.
Okay, so it seems like, as is typical in stroke, that there was a wide variety, a lot of variability in the muscle volumes across groups as well.
Okay, I think we'll go ahead and take one more question, and this is from Sarush Bagheri. Did you consider any crosstalk effects when you measured the EMG and compared it to your simulation results?
Nope, that's an easy one. No, we didn't. It's very possible that we had some, but the EMG is quite variable to begin with in this population, so I'm not quite sure how well we'd we would tease that out versus what's actually happening that may be abnormal as well. So while we didn't, it's probably worth looking into. We just didn't do it here in this case.
Okay, how much do you think that would affect? I mean, the main thing that you were looking at with the EMG was the timings. How much do you think any crosstalk could have affected the timings that you analyzed?
I think it certainly could a little bit, but the ranges that we were looking at as far as how well they agreed, I think there would still be the trend of an increase in agreement. We were increasing 10, 15%. When we use the subject-specific parameters, I still think that trend would follow.
Okay, great. All right, well, I'll go ahead and we'll close the question and answer session. Thank you, everyone, for the really great questions. That was a great discussion. A few closing remarks. OpenSim and this webinar series are supported by several grants from the NIH and the EU, including an NIH grant that funds our National Center for Simulation and Rehabilitation Research. We're also supported by the DARPA Warrior Web effort. Again, I really want to thank everyone for the great discussion and thank our speakers for a great talk. You can find information about the NCSRR and other upcoming events like future webinars and other resources on our website, opensim.stanford.edu. And also, please complete the survey that will appear in the pop-up window at the conclusion of the webinar. This will help us improve our webinar series and also choose upcoming topics. So again, thank you everyone for participating, and we hope you'll continue to stay involved with OpenSim.